I'm Paul Noble from Duke University, and my co-chair here, Rosa Schlenker-Herzeg from Boehringer Ingelheim, would like to welcome you to the last session at what has been a very successful meeting. We apparently don't have any additional housekeeping issues to discuss, so we'll get right into the science. And who better to start with than Victor Thannickal from the University of Alabama in Birmingham. It's going to reconcile some IPF paradoxes for us.
Thank you very much, Paul, and thanks to Luca and the group here for organizing this really wonderful meeting. I'm not sure I can reconcile all the paradoxes, but I hope that at the end of this we can at least raise some good questions about some of the difficulties we face in tackling this very, very difficult disease. So I'm defining paradox in a very broad sense, observations or statement or a group of statements that are not easily explained, that don't make sense, and may not really be inherently congruent. And there are several of those in IPF. There is the apoptosis paradox that Jeff Horowitz talked about on the very first day, where alveolar epithelial cells seem to be undergoing increased rates of apoptosis, whereas mesenchymal cells seem to be resistant to apoptosis. And then there is this matrix paradox, and I think you'll hear a little bit about this later on with Annie, where despite the fact that there is an increased expression of MMPs in the IPF lung, there seems to be a problem with matrix degradation. And there's also this very unusual localization of these fibrotic regions in IPF, where most of the fibrosis is in the periphery and in the bases of the lung. And does that simply relate to bioactive components of the matrix, or does it involve certain other factors like biophysical signaling and tension? And then there is a therapeutic paradox that we all talk about, where even though a number of agents have been proven to be effective in animal models, in patients they just don't seem to work. And how about an aging paradox? Even though we all recognize that IPF is a disease of aging, there just doesn't seem to be a very good explanation as to why that is from a cellular standpoint, whether you look at the mesenchymal phenotypes or the epithelial phenotypes. And then if you compare the animal models and the human scenario, there seems to be some disconnect in trying to explain aging, cellular senescence, and IPF. And there may be others. So I'm going to talk briefly about the matrix and some biophysical components signaling from the matrix, and touch a little bit on the aging paradox and some animal models that may be more relevant. And then maybe that will inform some of the issues related to therapies and apoptosis. So this is some work done by a junior faculty member in our division, Yang Zhu. He's really a bright young guy who's doing some work on biomechanical signaling. This was published in the Red Journal just last month, where he showed all in vitro, but that stiffness of the matrix can drive an intrinsic mechanotransduction where you have polymerization of globular actin, which when it polymerizes to filamentous actin, releases a transcription factor called MKL1 or MRTFA, also known as myocardium-related transcription factor, translocates to the nucleus and along with SRF or serum response factor binds to CARG elements in certain genes that drives the transcription of these genes, including alpha SMA and COL1. And since this is published, I'm not going to go over this data, but I'm going to show you some new data related to what Yang is working on, where he expands this paradigm to involve an anti-apoptotic pathway where the tension-induced or the polymerized actin dependent release of MKL1 leads to BCL2 expression and the induction of an anti-apoptotic phenotype through the mitochondrial intrinsic pathway. And he's shown this not only in vitro, but also in vivo. And I want to suggest to you that with this pathway, by blocking the Rho kinase pathway, that you might be able to modulate fibrogenesis and potentially even reverse it or arrest the progression of fibrosis. So this is some in vitro studies showing that when you culture IPF fibroblasts, and when you do this on in tissue culture, you do see these well-formed actin stress filaments that defines a myofibroblast. It's probably the best definition, not just alpha SMA expression, but the fact that they have this contractile phenotype with this very well-formed filamentous actin. The control fibroblasts, which were from failed donors, don't have that, even though they may express some alpha smooth muscle actin, they don't have those stress fibers. And what's interesting is when you treat the cells with Fascidil, you get a clear induction of apoptosis in any of these cells that have these well-formed actin stress fibers, so-called myofibroblasts. And you don't get the apoptotic response in the control fibroblasts, and this is quantitated here. Within 24 hours, you get a nice induction of apoptosis with Fascidil, but in the control fibroblasts, you do not. And then in vivo, in the explained and widely used, I'm not sure it's well explained, but widely used bleomycin model, when you treat the mice with Fascidil at 25 milligrams per kilogram on day 14, so post-inflammatory, and evaluate the lungs of these mice at 28 days, so a 2-week treatment of these mice with Fascidil, and you look at the frozen tissues for tunnel assays, you do see apoptosis in both scenarios, both in the saline-treated mice and in the Fascidil-treated mice, but the pattern of apoptosis that you see is quite different. So in the PBS-treated mice, you see that the tunnel-positive cells, the apoptosis is primarily in these lining cells, the alveolar epithelial cells, whereas when you give Fascidil, the apoptosis is in the interstitium and specifically in those cells that have alpha smooth muscle actin. So this is sort of a reversal of that apoptosis paradox where you get, in injury, you get epithelial apoptosis and mesenchymal resistant apoptosis, where Fascidil seems to reverse that phenotype. And if you, we started to look at pathways that lead to this apoptosis-resistant phenotype in IPF fibroblasts, and you can see here that when you treat cells with Fascidil in a dose-dependent manner, there's a downregulation of BCL2. This is at 24 hours by real-time PCR, whereas some of the other anti-apoptotic BCL2 family members, such as BCLXL and MLC1, are unaffected. And the induction of apoptosis in these cells, as is consistent with the BCL2 family, is related to the intrinsic mitochondrial pathway, where treatment of cells with Fascidil leads to a time-dependent release of cytochrome C from the mitochondrial inner compartment to the cytoplasm. You can see this with these assays where you see the expression of cytochrome in the mitochondria being reduced and an increased release of this into the cytoplasm. And with assays for caspase 9 and 3, which are also part of the intrinsic pathway, you see that there is induction of cleaved caspase 9 with Fascidil as well as caspase 3. And by ELISA, you see that, again, the intrinsic pathway is activated. Now, very different from what happens in the IPF fibroblasts, in the normal fibroblasts, you don't see this response. And this is just to show that there is no release of cytochrome C from the mitochondria to the cytoplasm. In fact, there shouldn't be any cytochrome C in the cytoplasm, and I found out later that this was we were trying to figure out what the expression levels were, and this was overexposed for over 24 hours in order to get any level of expression of cytochrome C. And you can see here again with caspase 9, 3, and the expression of all of these BCL2 family members, there was no change. Now, what is the mechanism for activation of this pathway that leads to the activation of the intrinsic mitochondrial pathway? So one of the mechanisms we had discussed earlier was this idea of the release of MKL1 once actin gets polymerized, and this is showing that you have a lot more F-actin or polymerized actin in IPF, and you can see that clearly and quantitated here as well. And along with that, this baseline higher level of myofibroblasts and polymerized actin, you see that there is increased activation or compartmentalization of MKL1 from the cytoplasm to the nucleus. And when you treat the cells with Fasudil, you see that there is some decrease in the amount of F-actin in normal cells, but a much more dramatic decrease in the IPF fibroblasts. And along with that, importantly, is the reversal of that cytoplasmic nuclear shuttling of MKL1, where this again now is retained in the cytoplasm with the treatment with Fasudil. So how does MKL1 transduce the signals to downregulate BCL2? So we generated a number of mutants of the BCL2 promoter, and you can see with the wild type that there is constitutive promoter activity in the IPF cells. And with the B1 mutant, which is missing this upstream CARG binding, or actually it's the B1 is the proximal and the B2 is the distal. But with the B2 mutant, you lose the constitutive BCL2 promoter activity. And in the cases where you have the constitutive induction, Fasudil clearly drops the promoter activation. And this is showing that the MKL1 SRF binding through CHIP assays or chromatin immunoprecipitation, showing that the MKL SRF complex binding to the BCL2 promoter is again inhibited with Fasudil. And then we did a number of studies, again, looking at actin dynamics in the absence of Fasudil when you use reticulin B, which is a compound that will depolymerize actin. You lose the expression of BCL2. And another compound, CCG, which is an inhibitor of the binding of MKL1 to SRF, again suppresses the expression of BCL2. An empty vector does not, but a dominant negative MKL1 does inhibit BCL2 expression. And this is in the scenario where you treat cells with Fasudil. You can overcome that inhibition with using this Jazz compound, which does the opposite of latriculin, which is to actually induce polymerization. So in the presence of Fasudil, polymerization of actin rescues this phenotype, and again, as does a constitutively active MKL1 that will again overcome the inhibition with Fasudil. And then looking at the other side of the coin, if you will, where again this is an appreciation for this whole pathway for myofibroblast differentiation on stiff matrices, just not the soluble factor itself transducing the signal from the TGF-beta receptor, but downstream there may be pathways that can induce myofibroblast differentiation, and this is simply showing that with TGF-beta on a soft matrix, which is 0.5 kilopascals, versus a stiff matrix, which in this case was at 20 kilopascals, you get an additive induction of the alpha-synuclein mRNA with real time, and that correlates nicely here with a Western blot showing that the synergistic induction of alpha-synuclein expression, both protein and mRNA, are inhibited with Fasidel. And this is looking at the morphology of those cells, both on soft and stiff matrices in the absence and presence of Fasidel. And you can see here that on the soft matrix, even with TGF-beta treatment, you don't get that clear myofibroblast phenotype, but there is very nice stress fiber formation on the stiff matrices in the presence of TGF-beta, and this is inhibited by Fasidel. Part of this myofibroblast phenotype is clearly the functional ability of these cells to contract gels. It's an in vitro correlate of what might be occurring in vivo. And you can see here that on soft matrices, you get induction of the contractile signaling, which involves phosphorylation of myosin light chain. And there is, again, similar to the alpha-synuclein studies, and synergistic induction of phosphorylation of MLC on the stiff matrices with TGF-beta, which correlates, again, with the gel contraction. And in both cases of treatment with Fasidel, you inhibit MLC phosphorylation as well as contractility. And this, again, looks at the actin dynamics component of it, where when you have alpha-smooth muscle actin induction in the stiff matrices with TGF-beta signaling, you're able to inhibit that in the absence of Fasidel when you depolymerize the actin stress fibers, or when you use a dominant negative MKL1, and here just the opposite experiments where you can rescue the Fasidel effects with constitutively active MKL1 or using the polymerizing pharmacologic agent. Now, what's happening in vivo? So both in the IPF tissues and in bleomycin-treated animals, there is activation of this pathway. So if you look at co-localization of where collagen is deposited in the IPF lung, you see that the activation of Rho kinase, and one of the downstream targets is the myosin phosphatase. You see that there is an expression of this, the phosphorylated form of MYPT in the areas where there's collagen deposition. And we couldn't use alpha-smooth muscle actin because of antibody cross-reactivity in these studies. But here with bleomycin, you see with alpha-smooth muscle actin, again, there is a localization of the activation of this pathway in regions of alpha-smooth muscle actin expression. Now, we went back and looked at the IPF fibroblasts as well as the non-IPF control fibroblasts, and you can see here that within ELISA that there is, again, constitutive activation of the Rho kinase pathway in these fibroblasts, as well as the fibroblasts isolated from the bleomycin-injured mice. And this also correlates nicely with PERM, which is a downstream substrate of Rho kinase, where you see again that there is constitutive activation of this pathway in IPF cells, as well as in the bleomycin injured fibroblasts in a constitutive manner. And this is using some other techniques to look at the Rho kinase pathway. This is using rhodokin beads, which binds to the active Rho A. And you can see here that with IPF, in the IPF lines, there is constitutive activation, whereas in the non-IPF, you don't see that. And this is true both in the animal model as well as in IPF. And then the important question of whether Facidal protects from experimental lung fibrosis. And again, this is treatment of the mice, as I'd explained earlier, at day 14 to day 27, and harvesting the mice on day 28. And you can see here that each of these points is a single mouse, and with bleomycin, you get a fibrotic response that is markedly inhibited in the presence of Facidal. And you can see here that whole lung homogeneous Fibrosis muscle actin is also downregulated with Facidal treatment. And we also showed that this downstream transcription factor pathway that's activated by the actin dynamics and polymerization of actin, MKL1 deficient mice are also protected from the fibrotic response to bleomycin, and this is quantitated here, and the wild-type mice here on the right, showing that you get nice induction of muscle actin as well as a fibrotic response that's inhibited with Facidal. Okay, so coming back to the pathway, so this is an entirely new pathway that Dr. Zhu has uncovered, where MKL1 transcriptional activation in contractile myofibroblasts induces the activation of BCL2 and activates an anti-apoptotic pathway, which is inhibited with Rho kinase. And he's shown this for the first time, that both in vivo and in vitro, with at least with the anti-apoptotic pathway, this is proof of principle that blocking the contractile response may be an effective strategy in IPF. All right, I'm going to shift gears a little bit for the last 5 minutes to aging, and I'll do this fast. So I think we all appreciate that this is a disease. This is a disease of aging, and we don't quite have an explanation for it. These are some hypotheses as to how epithelial cells may give rise to a fibrotic response. Clearly, there is a lack of regenerative response associated with activation of mesenchymal cells, and there are a number of questions that are not quite answered. There is this idea of cellular senescence being associated with aging, but it appears that at least in fibrosis, a lot of us think that proliferation is important, but really is it? And I think that's worth looking at carefully. And then this idea that has been published recently about the potential role of senescence in terminating fibrosis in other animal models. And as you've heard about earlier at this meeting, there are animal models that seem not to fit with what this association of fibrosis with aging. So these are two papers that were published recently, or fairly recently, showing that the acquiring of a senescent phenotype in fibroblasts may actually limit the extent of fibrosis. And there are some explanations for that which we can talk about. And again, these were done all in young mice, and were done with, as we do in the lung, with single injury. And let's go back to patients and look at IPF tissues and the fibroblastic foci and see what's going on there in terms of senescence and proliferation. So we actually did not see a lot of proliferation in these fibroblastic foci, and I think a number of groups are beginning to understand that. You do see some Ki67 positive staining in the epithelium and maybe in the periphery of these fibroblastic foci, but clearly these myofibroblasts are in the foci themselves and not undergoing proliferation. And you see increased expression of P16, which is a tumor suppressor gene that is involved with a cellular senescence that seems to be highly expressed in these myofibroblasts. And there is, as you would notice, a lot of P16 expression in the epithelial cells. And then if you look at apoptosis, and I think this other people have shown as well, Moses and Bruce U-Hall back in the room there, that there is this so-called apoptosis paradox, again, that Jeff talked about on day one, where epithelial cells in those apparently senescent cells are undergoing apoptosis, but the fibroblasts that are also showing markers of senescence don't undergo apoptosis. So we did the bleomycin studies in the age mice, and you can see here at the time of peak fibrosis, around 3 weeks, you get collagen expression with trichrome staining here. That doesn't seem to be a whole lot different. In fact, when we quantitated this in the age mice, it was actually less in the age mice, which was surprising to us. It was not, we didn't expect that. But when you looked at 2 months, there seemed to be more in the age mice than in the young mice. And then when we quantitated that, if you look at the time period between 3 weeks and 6 weeks and looked at the changes in total lung collagen, there was actually a decrease in the young mice, and this is about 15 to 18%, so there is some partial resolution, not complete resolution, but certainly partial resolution in the young mice, whereas in the age mice, there's absolutely no decrease, and in fact, a trend towards an increase in collagen between those time points. And if you look at the mice themselves, the young mice and the age mice lose weight. The young mice recover that by 4 weeks, whereas the old mice do not. And if you look at the lungs themselves, there is increased lung weights in the young mice that seems to partially decrease at 2 months, whereas this does not occur in the age mice. And this is showing that at 2 months, there is persistent expression of alpha-muscle-actin cells in the non-resolving age mice. And this is associated with the isolation of fibroblasts from these age mice that seem to have a persistent expression of P16 and beta-gal, which is a marker of cellular senescence, and this is quantitation for those cells. And then we looked at the phenotype of these aged and young fibroblasts in response to various apoptotic stimuli, and in response to H2O2, the age mice don't lose viability. This was the MTT assay, and this is with the caspase activity assay showing that the age mice are relatively protected from various forms of apoptotic injury. The age mice have higher levels of BCL2 at baseline, and lower levels of DNA fragmentation factor, which is in the pro-apoptotic pathway. And there is the increased expression of NOX4, which is a reactive oxygen species generating enzyme that we had previously shown is important for the fibrotic response. And what we are able to do is in this resolving period, give mice intranasal siRNA. We've done intratracheal as well. And we're able to downregulate the expression of NOX4 in the fibroblast. And this is an interesting phenomenon. It's a consistent finding, not only by our group, by others as well. And that we could talk about as well as to how that's happening. But when that happens, you're also able to downregulate profibrotic factors, downregulate the expression of P16 as well as BCL2. And this is associated, well, there you go.
Well, I think that's the message we got.
That's a beta-gal staining that showed that there was less, this is the MAC-PC problem, I guess. But this is the most important aspect of this, which is that the siRNA-treated mice during that resolving period you're able to reverse the phenotype and change the wild type, which is a partially resolving phenotype, to a resolving phenotype with the age mice. So this is what we had shown previously, and since we published this paper in 2009, I'm actually happy to say that a number of pharmaceutical companies have taken interest in NOX4 and are generating small molecule inhibitors to it. In fact, there's a phase 2 trial that is soon to be started by GenQtex, and they have a compound called GKT-137831, which is going to be tested in diabetic nephropathy. And the reason for this was because the initial interest was in IPF based on our studies, but they showed that similar pathways were activated in the kidney. And because the endpoints were easier with kidney fibrosis, they decided to take it for that indication. But as our story evolves on NOX4, what we believe may be going on is that in the young mice, there is this so-called transient senescence phenotype where the cells do undergo apoptosis and you can resolve the fibrotic response, whereas with aging, there is this sustained senescent phenotype and where the cells do become apoptosis resistant and you may get a persistent or progressive phenotype. So if you were at the Keystone meeting, you may have heard my discussion, which Paul thinks I talk too much, but about this evolutionary concept of adaptive responses and the fibrosis being a normal wound healing response. And in fact, as I mentioned yesterday, bleomycin I consider as a normal wound healing response. And we see this with ARDS all the time. Most of the patients that survive the first month, most of them live. When they live to 1 year, their lung function at 1 year is almost completely normal. So that is consistent with humans and mice being able to resolve injury. Aging seems to be associated with a non-resolving fibrotic response. We believe NOX4 mediates fibroblast senescence and apoptosis with resistance, and we're really excited about the possibility that modulating this intrinsic mechanotransduction pathway with Rho kinase inhibitors may represent an effective antifibrotic strategy. A number of other members of our group at University of Alabama in Birmingham that working on other aspects of fibrosis that I didn't get a chance to talk about, but Vina Anthony is working on pleural mesothelial cells as a potential precursor to myofibroblasts that maybe in 2 years you may hear about. I'm trying to get them to come to Hamilton possibly. And Gang Lu has worked on the microRNA 2131 more recently, where he's showing that it has an anti-fibrotic effect on modulating the myofibroblast phenotype. So I'll stop there. A number of folks in my laboratory and collaborators, people that I've worked with in the past, Jeff, who's done really well at Michigan, who I'm very proud to say is exceeding my own level of accomplishment in the field. So that's always good to see. Ragini, who is at Indiana University, and a number of collaborators, thanks, and I'm happy to take any questions.
Very nice talk, Victor. I'm interested in the apoptosis paradox, as you know, and most, if not all, of your very nice in vitro data were from myofibroblast. Mm-hmm. Yet I remember a slide you showed near the beginning where when you gave the fasudil in vivo, there was a shift from epithelial apoptosis over to myofibroblast apoptosis without epithelial apoptosis. So I'm wondering if you believe that the mechanisms that you've shown very nicely in fibroblasts in vitro are also happening in epithelial cells, or if not, are they the same, do you believe, or do the epithelials not express some of these signaling pathways? How do you explain the paradox and your observations with fasudil in vivo?
Well, that's a very intriguing observation, that you are able to reverse those apoptotic phenotypes in those two compartments. And one way to explain it is some of the work that you've done. So if you believe that fibroblasts or myofibroblasts, by factors that they produce, can induce epithelial cell killing, apoptosis, that is one potential mechanism, because we believe that the that the Rho kinase inhibitor is working primarily on the myofibroblasts. So there are at least 4 different candidates. One is your favorite molecule, angiotensin 2 peptides, which have been shown, which you have shown very nicely to induce apoptosis. We have shown that hydrogen peroxide produced by myofibroblasts can induce apoptosis. I think a group in Boston has shown that the myofibroblasts secrete fast ligand, and myofibroblasts are a source of TGF-beta. So at least there are 4 candidates that with this paracrine signaling of myofibroblast to epithelial cells could account for the reversal of that phenotype.
Thank you.
Not yet.
Oh, here we go. Okay, can you hear me?
Mm-hmm.
Victor, my question's a little bit related. So you had nice in vitro data showing that inhibiting the ROCK pathway is anti-fibrotic via several mechanisms, apoptosis one of them, and TGF-beta induced differentiation another. There was a bit of a disconnect, I thought, between that and the in vivo. So it wasn't clear to me, A, what the key pathways were in vivo, or if you've investigated those, and then B, what was the main cellular target in vivo of your inhibitor?
Yeah, it was a great question. So the beauty of this pathway is that we think that this selectively targets contractile cells in which Rho kinase pathway is highly activated. So when you use any pharmacologic agent, you're going to get more of a selective response in those cells where that pathway is preferentially activated. So myofibroblasts have a constitutive activation of Rho kinase. With epithelial cells, I'm sure it may be important, but may not be as important. So you have a therapeutic window. And we believe that the target with Fasudil is on the myofibroblasts. And we've shown in vivo that that occurs because Fasudil induces apoptosis of those cells and not of the epithelial cells. In fact, the epithelial cells look as if they were protected. So I think that the in vivo and the in vitro data are congruent in this sense.
Okay, thank you.
Any last question?
Victor, that's really beautiful work. A comment and a question. So we've been, on the first part of the talk, we've been interested in similar pathways looking at how LPA and other things that regulate the actin cytoskeleton talk to the nucleus in terms of profibrotic gene expression. And at least in vitro, we see a fair amount of redundancy between MKL1 and the related transcriptional coactivator MTRFB or MTRFA and MTRFB. And I just think it's very interesting. It looks like in vivo, MKL1 is really having a dominant effect. And I wonder if you've looked at all the possible role of MTRFB. And then secondarily, do you have any data that would point towards ROCK1 or ROCK2 as the most important isoform of ROCK that you're inhibiting in this pathway?
We have not looked at MRTFB, and Fasudil is not isoform specific.
Right.
So I think it's going to inhibit both pathways, but ROCK1 may be more important in terms of the signaling in the fibroblasts, but it could be inhibiting both isoforms.
Great.
Thank you.
So given that myofibroblasts in theory could come either from fibroblasts or epithelial cells, at least for your in vitro experiments, and this is related to Jeff's question, if you treated epithelial cells with TGF-beta to induce a myofibroblast phenotype, would those cells behave the same way? Because in your in vivo model, you don't see many epithelial cells that express SMA, but I'm saying if you were to induce that phenotype, would you be activating the same pathway, i.e. are myofibroblasts derived from different origins behaving similarly?
I think that's a very good question as well in terms of what the conserved pathways of TGF-beta signaling from fibroblast to myofibroblast differentiation and from epithelial to a mesenchymal phenotype. And we have observed that there is a lot of similar pathways. And certainly in the cancer literature, in EMT, the Rho kinase pathway is activated. So there again, if you argue that side, that EMT is important, then this strategy would be protective from mediating EMT and a fibrotic
except
response.
that you may then get an apoptotic response depending on if EMT is occurring and depending on what you believe it's doing. The reason
Right.
that I asked the question is because if you look in IPF lungs, as you know, you see, regardless of what you call it, I don't like the word EMT anymore, I think
Okay.
the process, but if you see epithelial cells that express mesenchymal markers, which you do, I don't know that they're necessarily in a fibril-associated pattern, but would those cells be susceptible to that pathway? And if you inhibited that pathway, what would happen? So I just put it out there as a
Yeah.
theoretical question because we actually had to quantify epithelial cell mesenchymal expression for our original EMT paper, and about 80% of those epithelial cells did have evidence of expression of alpha SMA. I don't know the extent to which this pathway would be expressed in those cells, but it's probably worth looking in the epithelial cells that express mesenchymal markers to see if this pathway is activated.
Yes, and more importantly in the IPF.
Yeah, in IPF
tissues.
lung, in IPF tissue.
Okay.
Thank you.
Thank you.